I'm going to talk on the chapter UV visible spectroscopy. So UV visible spectroscopy is one of the important spectroscopic techniques used in the organic chemistry to determine the unknown structure of the compound. Generally, UV visible spectroscopy is used for determination of the unsaturation or conjugation present in the organic compound. There are various types of spectroscopic techniques that are used in the organic chemistry, particularly like NMR spectroscopy, IR spectroscopy, UV visible spectroscopy, mass spectroscopy. So these spectroscopic techniques are really very useful in the research field because in organic chemistry, if we take the research field, various types of new organic compounds are synthesized, and we have to know the correct structure of this newly synthesized organic compound. So if we have the tools of this spectroscopic technique, then we can determine the unknown structure of that newly synthesized organic compound very easily and in very short period. In previous times, there were no such type of spectroscopic techniques, and it takes a lot of long period to determine the structure by carrying out simple chemical reactions only. But now the one of the advantage of this spectroscopic technique is that the compound is not destroyed or destructed. So these techniques are non-destructible techniques except the mass spectroscopy where the sample is destroyed by the bombardment of electrons, but all other spectroscopic techniques where we generally use electromagnetic radiation, and those techniques are non-destructive techniques. We can recover the sample organic compound after the successful spectroscopy. In this particular slide, we can see the electromagnetic radiation and the spectroscopic techniques associated with this. We know that there is a broad electromagnetic region or broad electromagnetic spectra in which radio waves, IR waves, UV visible waves, X-rays, gamma rays, microwaves, all these kinds of electromagnetic radiation is present. Now if you see here the radio waves that is used in the NMR spectroscopy and the changes that are taking place in the molecule, that is the nuclear transitions. So nuclei can be excited from one energy level to the higher energy level by absorbing the radio electromagnetic radiation. If you see here infrared waves or infrared electromagnetic radiation that is associated with IR spectroscopy. By absorbing the IR waves in the compound, the vibrational changes take place in the molecules and different types of vibrational levels can be created. So the molecule can be transferred from one vibrational energy level to the another vibrational energy level. But in this UV visible waves, they are related to the UV visible spectroscopy. We are using this part for the UV visible spectroscopy. By absorbing the UV visible light, electronic transition take place, means electrons can be excited from the ground energy level to the higher energy level. Both ultraviolet and visible spectra are also called as electronic spectra, as both are caused by electronic transitions. Now if you see here the range of visible electromagnetic radiation, U visible electromagnetic radiation is the one part of the broad electromagnetic range. Further, we can divide this UV visible electromagnetic radiation into two parts, three parts. One is far UV region, that is about between 50 nanometer to 200 nanometer. Another is the near UV region between 200 to 400 nanometer. The third one is called as visible, that is from 400 to 800 nanometer. Now this far UV region where the wavelength is very shorter wavelength, and this particular part is not useful for the UV visible spectroscopy because that cannot be detected by the spectrophotometer. Therefore, we cannot use this far UV region. We can use only two parts. One is near UV between 200 to 400 and visible region, that is 400 to 800 nanometer. So these two parts can be detected in the spectrophotometer, and therefore we can use, or these two parts are very useful in the UV visible spectroscopy. Now this visible region means here between the range 400 to 800, our eyes can detect this part. That's why it is named as visible region. We know that the ordinary light is composed of the 7 colors, that is VIBGYOR, and this part is related to the visible region. You have to also remember this important relation that is called as 1 nanometer is equal to 10 raised to minus 9 meter. This relationship is very important. Another relationship is between the energy and the wavelength. If the wavelength is shorter, then energy will be very higher, and if the energy is wavelength is longer, then energy will be very less. So this inverse relationship between the energy and wavelength, that should be remembered. Another important thing about the frequency and the energy: if the frequency is higher, energy will be also higher. If the frequency is lower, then energy will be also lower. UV visible electromagnetic radiation is highly energetic enough to bring about electronic excitations from lower energy level to higher energy level, that is electronic transitions. So these electronic transitions take place in UV visible spectroscopy. Therefore, it is also called as electronic spectroscopy. Electrons can be excited from lower energy level to the higher energy level by absorbing the UV visible radiation, and that's why it is also called as electronic spectroscopy. Now, if we see here the meaning of spectroscopy, generally we are using electromagnetic radiation for the different types of spectroscopic techniques. This electromagnetic radiation interacts with the matter and we get the spectra. Now if you see the example of prism, when you pass the ordinary sunlight from the prism through the prism, then this sunlight get split into 7 colors, or we get the spectra of 7 colors. So after interacting with the prism, there is one part, that is matter here, we get the spectra, and this spectra is useful for determination of the structure of that compound. We get some important information about the structure of that compound. So basically we can say that spectroscopy is nothing but the interaction of the electromagnetic radiation with the matter. Now in spectroscopy, there is measurement and interpretation of electromagnetic radiation absorbed or emitted when the molecules or atoms or ions of sample moves from one energy state to the another energy state. So when this electromagnetic radiation interacts with the matter, you get the spectra, and that spectra has to be analyzed, and after analysis you get the information about the structure of that compound. Now when this electromagnetic radiation is absorbed, some changes take place in the molecule or atom. Generally, suppose if you consider here usable spectroscopy, electronic excitation take place. In case of NMR spectroscopy, nuclear transitions take place. In case of IR spectroscopy, the changes in the vibrational energy level take place. So these changes are taking place, and after that we get the spectra, and that spectra gives the information. The usable spectroscopy, there is a principle, and that principle is nothing but the absorption of the ultraviolet visible region by the molecule or by the atom or by the compound. And due to this absorption, there is excitation of the electrons from the ground energy level to the higher energy level. So you have to see that in invisible spectroscopy, the electromagnetic radiation is in the range of UV and visible range or waves. This particular part we can use for the usable spectroscopy. Then that can be used for determination of the structure of the compound. So it particular part get absorbed by the molecule, and when it is absorbed, electronic transitions take place. So that light, UV visible light, get absorbed by that compound, and when it is absorbed, the electronic transitions take place. So that is the basic fundamental principle behind the UV visible spectroscopy. So before studying this UV visible spectroscopy, we have to also know about this Beer-Lamberts law. That is the basic laws that are very important, or we can say the foundations of the usable spectroscopy, on which the spectrophotometer, that instrument, works. So Beer-Lamberts law we have to learn, and that is very important. Now let's see here. This is the vessel containing the compound having the concentration C, and it is the length is L. Now if we suppose here incidental light of the intensity I0, and when it passes through the solution having the compound with the concentration C, after passing this light you get here transmitted light, and it will have the intensity I. So the intensity of the incident light and intensity of the transmitted light is not the same. They are not the same. They are different. So here actually there is a decrease in the intensity of the transmitted light. And this decrease in the intensity of the transmitted light depends on the concentration of the compound present in the solution and the length of this absorbing medium. And on these two factors, the intensity of the transmitted light depends. So the first law, the Lamberts law, which can be stated as when a ray of monochromatic light passes through an absorbing medium, its intensity of light decreases exponentially as length of the absorbing medium increases. So this intensity of the incident light decreases as the length of the absorbing medium increases. So if we increase the length of this absorbing medium, then there is exponential decrease in the intensity of the transmitted light. So this Lambert's law can be mathematically written as I, that is the intensity of transmitted light, is equal to I0, that is intensity of the incident light, e raised to minus k1 L. L is here the length of the absorbing medium. Then the next law is Beer's law. When a ray of monochromatic light passes through an absorbing medium, its intensity of light decreases exponentially as the concentration of the absorbing solution increases. So if we increase the concentration of that solute present in the solution, then it is found that there is a decrease in the intensity of the incident light. So this law can be also written mathematically as I, that is the intensity of the transmitted light, is equal to intensity of the, that is incident light I0 e raised to minus k to c, where c is the concentration of the compound or solute present in the solution. Now we can combine these two laws, that is Lamberts law as well as Beer law, and it will become now Beer-Lamberts law. Now if we combine these two laws, that is these two equations 1 and 2, we get new equation. So the intensity of the transmitted light is equal to the intensity of the incident light multiplied by e raised to minus kcl. Now here we have taken k1 dot k2 is equal to k, so we get here new constant k. Now we can rearrange these equations and we get this following set of equations. Now here I upon I naught is equal to e raised to minus kcl. Then we take the log to the base e, I upon I naught is equal to minus kcl. So e is removed here. We have taken the log to the base e. Now if we move this I naught above and I below, then this negative sign get cancelled. So log to the base e, I naught upon I is equal to kcl. And finally, if we take the log to the base 10, I naught upon I is equal to k divided by 2.303 into c dot l. C is the concentration of the solute or compound present in the solution, and L is the length of the absorbing medium. Now from this equation we get this value, that is log to the base 10 I naught upon I is called as absorbance, that is denoted by A, capital A, absorbance, and K divided by 2.303 is called as extinction coefficient, epsilon. So we can write above equation as, so this equation can be written like this: A is equal to epsilon dot c dot L. Now suppose if you take the concentration of that compound 1 molar and length of the absorbing medium suppose 1 centimeter, then A will become equal to the epsilon, and that is nothing but the molar extinction coefficient. So here you have taken the quantity 1 molar and length of that absorbing medium, that is 1 centimeter. Then we can call it as a molar extinction coefficient. So epsilon will become equal to the A, capital A, that is absorbance. Now the UV spectra is recorded as a graph here, and that graph is of the absorption versus wavelength. So wavelength and absorption can be plotted on the graph. Wavelength is on the suppose x-axis and absorption will be on the y-axis, and you will get the curve according to the absorption of that light. You will get that curve, and from this you will get the spectra, and that is nothing but the usable spectra. So this Beer-Lamberts law holds good for dilute solutions. Generally the concentration is 0.01 molar and not over the entire range of concentration. So you have to be careful while choosing the concentration. It shouldn't exceed 0.01 molar. So below the 0.01 molar you will get the good results, and above this there will be some we can say distortions. The absorbing species should not undergo association, dissociation, or reaction with the solvent. This law is not obeyed if the radiation is polychromatic, and deviation may occur if the solution contains impurities. So these are some of the limitations we can say about the Beer-Lamberts law. Now in this particular slide, you can see the UV spectra of some compounds. So this is the compound here shown having the conjugated system, triple bond, single bond, and double bond. So conjugation is present here. And one of the important application of UV spectroscopy is to determine the nature of the conjugation present in the organic compound. Now you can see here the absorbance on the y-axis, then wavelength on the x-axis. And after this, you will get the here curves as a spectra. So this point here, you will find that the absorption is maximum. Absorption is maximum, and because the epsilon value is higher here, so absorption is maximum. So to this value, here you get the lambda value, and that lambda value is called as lambda max value, where the absorption is maximum. So by this, by observing this graph, you get the lambda max value, and this lambda max value is very important because for the compound we get only one lambda max value, and this lambda max value depends upon the structure of that compound. So here this is the structure of that compound, and so this lambda max value is associated with the nature of the double bonds or triple bonds present in the compound. Another example is here, this compound is you can say benzene ring double bond, then this carbonyl group, they are all in the conjugation. And so here also the lambda value that is on the x-axis, absorbance on the y-axis, and you get two lambda max values, but this is important lambda max value with the high absorption, there is extinction coefficient, this is low. So in case of this compound, this pi to pi star transition as well as n to pi star transition take place, that we will see later on, and therefore we are getting two lambda max values. So these lambda max values are very useful in the UV visible spectroscopy as they give information or some clues about the nature of the unsaturation present in that particular organic compound.